All right, so new topic. What we are talking about now is the events that occur at the neuromuscular junction. What does that mean? Well, this is going to be all of the events that occur when a muscle cell gets stimulated by a neuron. What I'm drawing with these big, long white lines is a muscle fiber. Same thing as muscle cell. Muscle fiber, muscle cell, same thing. We call it a fiber because it's long and skinny. So here's a skeletal muscle cell, and this structure that I'm drawing here looks like Bart Simpson's foot, is a axon terminal of a neuron. So I'm just going to write neuron, and if you haven't gotten to the nervous system yet, you will. We'll discuss a little bit about what the neuron does, mostly about what happens down here. So here's the motor neuron. Here is the skeletal muscle cell. I'm going to write skeletal muscle fiber, single cell. We call this connection point the neuromuscular junction. Note that there's no physical contact between the nerve cell and the muscle cell. That's how it is in life, in the human body. I've exaggerated this space a bit because we're going to draw some things in it. So this is the neuromuscular junction. We're going to discuss the events that occur at this junction, and then what happens inside of the muscle cell to make it contract. First, the nerve cell, the neuron, will send an electrical stimulus called an action potential down its axolemma, or down the membrane of the axon. So I'm going to draw an electrical stimulus. This little jagged line represents electricity coming down the neuron. When it gets to the stimulus, when it gets to the end of this neuron, called the axon terminal, this electricity along the membrane causes pores or channels to open up on the membrane. And calcium ions, extracellular calcium ions that are outside the cell, influx into the cell like this. So we have electrical stimulus, pores open up, or gated channels open up, calcium enters. These bubbles are called vesicles. They store neurotransmitter or chemical. In another lecture, we discussed when a nerve cell is at rest, it's just a chemical manufacturing plant. It just makes chemical and stores it in these vesicles or bubbles. So I'll label this neurotransmitter. That means chemical stored in a vesicle. That's this bubble. Now, another lecture which we're not going to get into right now is how calcium causes this vesicle to migrate to the end of the cell and release its chemical. That's a whole other lecture. What we are interested in now is mostly is what happens down here. So calcium enters, it causes these vesicles to migrate to the edge, fuse with the membrane, and release the neurotransmitter or chemical. So this picture that I've drawn right here represents chemical being released from the motor neuron, neurotransmitter being released, and diffusing across this synaptic cleft. Diffuses means it requires no energy. It's just chemical going from a higher concentration to a lower concentration. So we have diffusion of neurotransmitter across the synaptic cleft. Remember, the synaptic cleft is just this space created by the neuron and the muscle fiber cell. This indented portion of the muscle fiber is called the motor end plate. And there are receptors on the motor end plate that receive this chemical, this neurotransmitter. Now, in the case of a motor neuron stimulating a skeletal muscle, there's a very specific chemical that's released. It's called acetylcholine. And in this specific case, acetylcholine, this chemical, has a stimulatory effect on skeletal muscle cells. So, just to review really quickly in 10 seconds, electrical stimulus, release of chemical, acetylcholine, diffuses across the synaptic cleft. This chemical then is going to bind to these receptors on the motor end plate. So now we have chemical binding to receptors on the motor end plate. That causes a whole series of events, chain reaction to occur now in this skeletal muscle. So we've got electrical stimulus, chemical release, binding of the chemical, acetylcholine. That causes an electrical stimulus to occur, an action potential to occur along the sarcolemma or membrane of the muscle fiber. Electrical, chemical, binding, electrical. So we have an electrical stimulus now radiating around the edge, the sarcolemma of the muscle fiber cell. That electrical stimulus isn't doing the cell, this muscle fiber cell, much good unless it can be guided inward to the inside of the cell. So these blue structures that I'm drawing here are called T-tubules. They literally, they get their name because they look like T-shaped structures, but they literally guide an electrical impulse along the edge of the muscle fiber cell inward to the inside. So in blue I've drawn T-tubules. They are literally open pores on the edge of the muscle fiber cell that guide this electrical stimulus inward. So I'm going to draw that. Here's my electrical stimulus. Now it's going inside of the cell, guided in by a T-tubule. At the base of each T-tubule, here, there are two storage sacs. I'm going to color them in orange. There are two storage sacs associated with the base of each T-tubule. They have a long, funky name, but that's just language. It's not confusing, it's not difficult to understand, it's not complicated. The long name, scientific name of one of these sacs is called a terminal cisternae. Cisterna would be singular. If I add an E to this, it means more than one. So this is a terminal cisterna. It's a storage sac, and it holds calcium ions. This is one reason why calcium is so important in the human body, is we've used calcium to release neurotransmitter, acetylcholine. Now we're going to use calcium again to cause this muscle cell to contract. So when this electrical stimulus is guided inward to the base of the T-tubule, where we have two terminal cisternae storing calcium ions, we call this area, by the way, a triad, because it's two terminal cisternae plus one T-tubule. So at this triad, when the electrical stimulus reaches this point, calcium ions are released from the terminal cisternae. And this happens at each T-tubule base in the muscle cell. So we get calcium ions flooding the cell. That's just step 1, 2, and 3 in this whole story. So we have step 1 up here, 2, and now 3. Calcium ions are released. We're going to find out what those calcium ions do on the board. Actually, I'll just move this board over here. Just pay attention to, keep in mind that we have gone through all of this process to do this, release calcium ions inside of the muscle cell. Now I'm going to quickly magnify the inside of a muscle fiber cell a gazillion times and simplify it for you. drawing now are contractile proteins, myofilaments, inside of a muscle cell, inside of those myofibrils, those tubes that we find inside of a cell. This represents the M line. This horizontal pink structure that I'm drawing is the thick filament myosin. So it has a head on it, actually has multiple heads, but this myosin molecule, I'll label it, is just called the thick contractile protein or contractile filament. Associated with the myofilaments is another amino acid chain-based structure called actin. It's a contractile protein or filament. This occurs thousands, hundreds of thousands to millions of times in one muscle fiber cell. There's a long chain of this arrangement. Ultimately, the end of the story is going to be we want myosin. Myosin wants to reach up, grab actin, attach to it, bind to it, and pull it this direction toward the M line. So if my arm is myosin molecule, what a contraction is in a muscle cell is this myosin molecule is going to reach up, grab actin, and pull it this way toward the M line. If we do that over and over and over again, hundreds of thousands of times, a muscle cell does this when it contracts. You know that. It shortens like this. This is just at the microscopic level of what's going on. So we left our last drawing with calcium flooding the cell. What is the significance of that? Here it is. Myosin wants to reach up, bind to actin, and pull it in order for a muscle cell to contract. But when this muscle cell is at rest, myosin can't bind to actin. There's an active site. There's tons of active sites on this actin molecule. I'm going to draw a few where myosin can bind to it. But when this cell is at rest, a muscle cell is at rest, there is a grapevine-like structure wrapped around this actin molecule that's covering the binding sites. It's just a peptide, multi-peptide structure called tropomyosin. Tropomyosin is this grapevine-like structure. It's like a stick. And it wraps around actin and it covers these binding sites so that myosin cannot attach to it. Attached to tropomyosin are these little tabs, 3 or 4 peptide structure. So I'm going to draw in green chalk. I'm going to draw these little tabs attached to tropomyosin. These little tabs are called troponin. And this is the arrangement inside your muscle cells, yours and mine. The goal now is for tropomyosin to lift off of actin, exposing these binding sites so that myosin can attach to it and ultimately pull it toward the M line. Calcium that flooded the cell, the inside of the cell, in our last picture, our last story, binds with troponin. And when, usually in the human body, when things bind together, they change shape when we get binding. So we get a physical binding of calcium ions to troponin. This whole complex changes shape, and tropomyosin is literally lifted off of actin, exposing these binding sites. So when calcium binds to troponin, tropomyosin lifts off of actin so that myosin now can bind to actin. Myosin binds to actin, pulls it toward the M line. We call that the contraction power stroke of muscle contraction.